All right, Bio Buddies. Are you ready for one of the most exciting presentations of your entire lives? We're going to go ahead and go through the process of cellular respiration from start to finish. If you have any questions, make sure that you write them down, and we'll talk about it when I get back in class. Hey, we got to start out with some things that you already know, but just so we're all on the same page. You already know that cellular respiration is the process by which oxygen is used to break down food like glucose and release energy. And we're already hopefully familiar with our summary equation, kind of sort of like this. But what you might not realize is that this is only a summary equation. In reality, things go more along the lines of this puppy. We'll see why. We're adding some extra water later. For now, just take it on faith. Also, some review. You remember glucose, C6H12O6, can exist as a couple of different isomers. We can have a straight line, a straight chain glucose, somewhat like this. When we do, we can go ahead and number our carbons, starting with the carbon that makes up our carbonyl, our aldehyde up here, our number 1 carbon, and go straight down the line to number 6. But if we're looking at a stable compound of glucose, in most situations it takes a ring form, and to do that we can kind of sort of fold it. We'll take this hydroxyl off the 5th carbon, rearrange it, and bond that carbon to the oxygen in the ring. So now that the 1st carbon and the 5th carbon share that oxygen, we'll take the hydrogen that was originally with that hydroxyl group, we'll give it over to the 1st carbon, and we end up with something like this. We still have the same numbering system. So our number 1 carbon, our number 2 carbon, our number 3, 4, 5, with our 6 carbon kind of hanging out in the space there. Well, the process of cellular respiration can be broken down into several little sub-steps. And we're going to be looking at each of these reactions in turn. The first one that we'll come across is glycolysis. And although this really isn't a part of cellular respiration, it always has to happen before the rest of cellular respiration occurs, so we're going to go ahead and treat it as part of the process. Glycolysis, well, lysis means to break apart. Glyco, we're going to refer to glucose, so we're going to be breaking apart glucose. This occurs out in the cytosol with no special structures necessary, as a completely anaerobic process, so no oxygen is required. However, we look at the other stuff that's needed for glycolysis. We, of course, need a glucose molecule because that's what we're going to be breaking down. We're going to be looking for some electron carriers or hydrogen carriers, whichever way you want to call it. So nicotinamide adenine dinucleotide without its hydrogen, so it's going to be its oxidized form here, right? I'm going to refer to this as a NAD, just because it's easier to say. But since it doesn't have its hydrogen, we can call it a naked NAD. We're going to go ahead and do some phosphorylating, dude. So we're going to take some ADPs and a couple of spare phosphates. And we're going to need an energy source. So in order for this reaction to occur, it's not spontaneous. We have to get a little bit of energy put into it. But what's cool is we get some more energy out of it than we actually put into it. So we get kind of a profit. Generally speaking, glycolysis is going to be exergonic. Even though we're putting energy into it, we're getting more energy out of it than we put in. Products, we're going to end up with 2 smaller molecules. Glucose is going to be broken not quite in half, but 2 smaller molecules called pyruvate, formula down here. And our NAD is going to be reduced. We're going to take up that hydrogen, but remember, to take up that hydrogen, we always end up taking up a pair of electrons as well. And like I said, we get a profit of 4 ATPs here. Excuse me, a output of 4 ATPs, a profit of 2. It's like investment banking, right? You invest $2 and you get $4 back, you make a profit. Well, if you invest 2 ATPs and get 4 ATPs out of it, you get a profit of 2 ATPs. Now, what actually happens in glycolysis is that we're going to go ahead and use 2 molecules of ATP to isomerize and temporarily phosphorylate the glucose. We're going to take that glucose ring, we're going to go ahead and create an isomer that is linear instead of ring-shaped. And it's going to be in the form of fructose rather than glucose. Remember, fructose still has the same formula, C6H12O6, but all of the hydroxyls and such are arranged in a slightly different fashion. So imagine glucose taken as a linear form with the number 1 carbon and the number 6 carbon actually being attached to the phosphates that we took away from ATP. I'll show you a diagram here in just a sec. The fructose bisphosphate, or the fructose biphosphate, you can say it either way, is then going to be broken down with the help of enzymes into PGALs, phosphoglyceraldehydes. And I'm not sure if you remember what a glyceraldehyde looks like, but it has a formula that looks something like this. Imagine a 3-carbon compound, and we can go ahead and put hydroxyls over here and hydrogens here, and then we're going to make this an aldehyde. So to be an aldehyde, we're going to have a double bonded oxygen over here and a hydrogen over here, right? And these are all single bonds between our carbons. Okay. Well, since this is a phosphoglyceraldehyde, imagine a phosphate down here. So I'm going to go ahead and change this to a phosphate. We'll make an oxygen, double bonded to a phosphate. We'll put a double bonded oxygen here, oxygen, and an oxygen, and we'll leave a little ion here, and we'll put that extra hydrogen right there. Okay? Well, each PGAL then is going to eventually become oxidized. We're going to go ahead and release a hydrogen, and that's eventually going to be picked up by NAD. And the PGAL will eventually release its phosphate, and we can go ahead and rephosphorylate the ADPs that we started with, reform ATP. And when all this is happening, we can actually end up, when this is released right here, we can actually end up as an exergonic reaction, which will release energy to phosphorylate 2 additional ADPs into ATP. Now, all this is done completely and totally with enzymes. And we don't need any special structure. The molecules act as the substrate reacting with the enzyme, or acting in concert with the enzyme. So we're going to go ahead and look at these molecules here as substrate molecules. Thus, since we end up with ATP at the end, this is referred to as substrate level phosphorylation. We'll talk about other types of phosphorylation later. It's usually helpful to see this in a diagram or a flowchart form, so here goes. We're going to start out with our glucose molecule, our ring-shaped glucose, and we're going to go ahead and isomerize that. We're going to break this ring apart. We're going to stretch it out into a 6-carbon linear compound, convert glucose to fructose, to do this, we're going to have to put some energy into it. So imagine 2 ATPs worth of energy going into this system to cause this to occur. Of course, we're going to get 2 ADPs out of it. What happened to the extra phosphates? Well, they're going to be stuck on the end of that fructose molecule. Thus, we will have a phosphate on the number 1 carbon and a phosphate on the number 6 carbon. Thus, 1,6-fructose bisphosphate or biphosphate. Well, we're going to go ahead and crack this in half. We're going to go ahead and make P-gals out of this. But before we do that, we need to go ahead and separate a hydrogen out. So we're going to go ahead and remove a hydrogen here. And that hydrogen is going to get picked up by NAD to make NADH. And what we're going to be left with is just our P-gal, which will have 3 of our original carbons, 5 of our original hydrogens, and 3 of our original oxygens. But whatever happens on, to one half of the cracked molecule is going to happen to the other half as well. So we're going to go ahead, do the same thing over here, strip off a hydrogen. It has to get picked up by NAD, and we're going to end up with NADH, and we'll have our P-gal over here. Right. So first step, we're going to go ahead and take glucose, break it apart, and get P-gals. All right, clean that up a little bit, because you know me, when I write, I sometimes get a little bit messy. So let's work on our P-gals. If we can phosphorylate something, then we should be able to dephosphorylate something. So imagine us taking this phosphate away from our glyceraldehyde. Well, we're going to have to go ahead and do that with the appropriate enzyme. When we do, though, we actually end up with another exergonic reaction. Enough so that when this phosphate is released, we can go ahead and allow ADP to swing over, pick that up, and become ATP. And as a bonus, we're getting enough energy released to convert another ADP plus a free phosphate into ATP. And lo and behold, what we end up with is something called pyruvate. Now, pyruvate looks a little bit like a P-gal without the hydrogen, and we don't necessarily have to worry about the exact molecular structure, but if you're just counting carbons, hydrogens, and oxygens from the original glucose, this pyruvate is going to end up with 3 of our original carbons, 5 of our original hydrogens, because remember we stripped off a hydrogen up here, and we'll still have 3 oxygens. Right? And we're, again, whatever happens on this side is going to happen on this side. So what we eventually end up with is something like this, all cleaned up. A couple things worthy of note. Once again, if we're looking at what we have left over from the original glucose, this pyruvate has 3 carbons, 5 hydrogens, 3 oxygens, And there's another one on this side, so we can multiply all this by 2. And we still end up with 6 of our original carbons, now 10 of our original hydrogens, 6 of our original oxygens. So most of it is accounted for. We're missing a couple of hydrogens because we started out with 12. Well, remember that one of those hydrogens is being carried by this NAD, and the other one is being carried by this one over here. Don't forget about those. They're not waste. They're going to be important later. Also, we had to invest 2 ATPs worth of energy, but we get 2 plus 2 over here in return. So we're getting 4 out of this, meaning we got a net profit of 2 ATPs, which is good. Always nice to have more energy, but the unfortunate thing is a profit of 2 ATPs worth of energy is not enough to keep complex multicellular organisms alive. So we're going to have to harvest a lot more energy. Luckily we can, because there's a whole lot more energy to be harvested. Okay, now the next set of reactions actually begins the true process of cellular respiration. And although we're going to call this next set pyruvate conversion is actually a portion of what's called the tricarboxylic acid cycle, or the citric acid cycle, or the Krebs cycle. Krebs cycle because it was discovered by some guy called Hans Krebs in the 30s. And citric acid cycle because citric acid is one of the components of the cycle, which we'll look at later. Or tricarboxylic acid cycle. Well, we'll see why it's named that in just a sec. But although this reaction is cyclical in nature, this portion of the reaction is not quite cyclical. So we're going to go ahead and take this out and look at this as its own separate series of reactions, even though it's not. Okay. So going into this pyruvate conversion, we're of course going to start out with a couple of pyruvates because that's what we ended up with in the last set of reactions in glycolysis. We're still going to be stripping off some hydrogens, so we're going to need some NADs. And we're going to need a carrier molecule to bring our products into the next series of reactions, the true cyclical portion of the reactions. And that carrier molecule is something called coenzyme A. So products, well, we're going to get rid of some CO2 as a waste. And you're supposed to have CO2 as a waste in cellular respiration, so this is the first time we're going to do it. So we're going to start stripping off carbon molecules from that pyruvate. And the rule of thumb is that anytime we strip off a carbon, we have to strip it off in the form of a carboxyl. Remember carboxyls? Q groups? COOH, right? So this is sometimes referred to as the tricarboxylic acid cycle because each of the pyruvates are going to have to lose 3 carboxyls. You'll see it in a sec. Anyway, once we strip off carboxyls, we no longer have a pyruvate. We're going to end up with something called an acetyl group. I'll show you that in just a sec. And that acetyl group latches onto the coenzyme A, or the coenzyme A latches onto the acetyl. So temporarily we'll have acetyl coenzyme A. The hydrogen that was once part of our little Q group, our little carboxyl group, actually gets picked up by NAD. And again, we'll see that happening. So in a nutshell, we're going to go ahead and strip off a carboxyl from each pyruvate. Each carboxyl is going to be oxidized in such a way that CO2 is just going to be released as a waste, and hydrogen is taken up by NAD, and that remaining 2-carbon fragment, that acetyl group, is going to be carried off by coenzyme A as acetyl coenzyme A into the actual cyclic part of the reactions. So to do this right, I'd have to go ahead and do this twice, but in your mind's eye, just think that, hey, this is happening to each pyruvate molecule, and we've had 2 pyruvate molecules. So I'm just going to double everything as we go. Right? So each pyruvate is going to lose a carboxyl. How does that happen? Well, you just need the right enzymes once again. So a Cu group, a carboxyl group, is going to get stripped off, but it's going to happen from each pyruvate, so we've got to put 2 of these in there. And we're going to go ahead and strip off the hydrogen. The hydrogen is going to be released. We're left with CO2, right? And of course there's 2 of these, and there's 2 of these. And this is just a waste, no biggie. But our hydrogens have got to be picked up by NAD. And of course there's 2 of those because there's 2 hydrogens. We pick up the hydrogens and we end up with 2 NADs. And what we end up with, instead of a pyruvate, since we've lost all this carbon, these oxygens, and this hydrogen, we end up with a compound that has only really 2 of our original carbons, now 4 of our original hydrogens, and just a single oxygen. But of course there's 2 of these. And I'm not going to go through the exact chemical formula, but we're going to call this puppy an acetyl group. So our cleaned up version looks something like this. And I'm going to continue to put our original glucose, or fragments of original glucose, in green so you can track them. And then anything that is given off as a product, I'm going to put in red. Okay. So we've stripped off our Cu, we've got our CO2, our missing hydrogens are here, and now we need to go ahead and carry this acetyl molecule into the next set of reactions. We need a carrier molecule to do that. This truly acts as just a carrier molecule. It does not really do anything to our acetyl group, at least nothing that we have to worry about. So we're going to go ahead and put in some coenzyme A. And of course there's going to be 2 of these because there's 2 acetyl fragments. And we'll end up with, at the tail end of this, 2 acetyl coenzyme A's. Now we're still going to have the exact same amounts of carbon, hydrogen, and oxygen that we have in a regular acetyl. It's just going to be carried by the coenzyme A. And there we have our cleaned up version of what has happened so far. Our acetyl group is going to go ahead and be dumped into the next set of reactions, which are indeed cyclical in nature, and basically a cascading series of reactions is going to allow us to go ahead and break apart what's left of our original glucose and destroy it completely. And we'll get a little bit of energy along the way. So this next set of reactions gets a little bit sporty. Even though pyruvate conversion is technically a part of these reactions, it's a pretty linear reaction. Now we'll see the cyclical portion. So again, Hans Krebs figured this out in the 30s, got a Nobel Prize for it. It's sometimes called the citric acid cycle for reasons which you'll see here in a sec, or the more descriptive tricarboxylic acid cycle, because once again, anytime you strip off carbons in this process, you have to strip them off as a carboxyl group. Right? So this also occurs in the membranes of the mitochondria. We're going to take our acetyl coenzyme A and dump it into the reaction. We're going to strip off hydrogens and have those hydrogens carried away. So we're going to need some hydrogen/electron acceptors. And we've been looking at NADs. And, we've got 8 hydrogens total in here, but only 6 of them are going to be carried off by NADs. The other 2 by FADH. We'll call them FADs. We're going to need some oxygen here. And technically, the Krebs cycle is anaerobic because it doesn't really use free oxygen. Instead, it uses oxygen that is stolen from a water molecule. And this is also important. The water is also going to be important because, remember, anytime we're getting NADs and FADs to accept a hydrogen, they have to be reduced. We're going to need electrons to actually go ahead and cause that to happen. Electrons don't just appear out of thin air. They got to come from somewhere. Well, we're going to use the hydrogen in water as an additional source of electrons here. So, yeah, we're going to need some water in here. What are you going to end up with? Well, once again, our NADs and FADs are going to be reduced, and they're going to be carrying not only the extra electrons, but the hydrogens that are released from that acetyl group. So we'll end up with 6 NADH and 2 FADH2s. We're going to strip off the remaining carbons as carbon dioxide, and we'll actually get a little ATP out of it. Again, not a whole bunch, but a little bit. And we're going to end up with those 12 pesky hydrogens from that water, or those water molecules, and we'll find out what happens to them later. Wouldn't this be a great short answer question? Describe the events of the citric acid cycle in detail. If you want to do this in paragraph form, you can do this by kind of sort of relating the following info. That acetyl coenzyme A is going to participate in this set of reactions, but coenzyme A just carries that acetyl group in. It's removed, and that acetyl fragment joins with a 4-carbon acid called oxaloacetic acid. Now, that oxaloacetic acid gets that extra 2-carbon fragment to form a 6-carbon acid called citric acid, hence the name citric acid cycle. Well, citric acid then is going to be broken apart. Not entirely. We're just going to go ahead and take bits and pieces of that original acetyl fragment and get rid of them. So one of the problems is that carbon can only be removed as a carboxyl group. But if you remember, each acetyl group has got 2 carbons, 4 hydrogens, and only 1 oxygen. We don't have enough oxygen to make a carboxyl group. So that's why we put some water in there. If we remove 2 oxygen atoms from a couple of water molecules, we now have enough to make 2 CO2s, or 2 carboxyls, I should say. We can strip off the hydrogen, let NAD take care of that, 2 CO2s as a waste. And now instead of a 6-carbon citric acid compound, we have a 5-carbon acid called alpha ketogluteric acid. Yeah, right. Well, we still need to go ahead and reduce that. Alpha ketogluteric acid has 1 of our original carbons, 3 of our original hydrogens, and none of our original oxygens. We stripped all those out. So if we want to go ahead and get rid of this carbon, we end up with, again, a problem of not having enough oxygens to make a carboxyl group. So we have to go ahead and add some more water, and this time it's going to take 4 molecules of water to get enough oxygen to make a couple of carboxyls. And once we get rid of another Cu group, alpha ketoglutaric acid is converted to succinic acid. Again, we get rid of CO2 as a waste, and the hydrogens from the Cus are going to be taken up by NADs. But what's cool about this is that when we go from alpha ketoglutarate to succinate, when that last carbon is released, it's exergonic. And we have enough energy to go ahead and phosphorylate a couple more ADPs into ATPs. So we get a little bit more energy out of it. The problem now, of course, we still have some extra hydrogen left over. So succinic acid differs from our original oxaloacetic acid because it's still got a couple of hydrogens, or extra hydrogens. So we're going to strip off one of those hydrogens from each of our fragments. And this time, FADH is going to be our reducing agent. Pick up that hydrogen. And we're going to convert succinic acid to fumaric acid. Fumaric acid looks really like oxaloacetic acid, except it's got one more hydrogen, the hydrogen from the original glucose. So we need to go ahead and take that hydrogen away, and we're going to use that to help reduce NAD and NADH, and we're back with oxaloacetic acid. So easier to see in a flow chart. So let's look at like a really cool flow chart. Okay, now here's where we left off. Up here, we had our acetyl fragment, two acetyl fragments, being carried by coenzyme A into this next set of reactions. Right? Each of the fragment, 2 original carbons, 4 original hydrogens, 1 oxygen. Well, our coenzyme A gets stripped off, and we're going to let each of these fragments join to a 4-carbon compound called oxaloacetic acid. Now I'm going to just leave this as a 4-carbon chain. It's got other stuff attached, but we're not going to deal with all that other stuff. We're going to be more concerned with what happens to these atoms in the carbon, hydrogen, and oxygen in that acetyl group. Well, once the acetyls join with oxaloacetic acid, you're going to have a 6-carbon compound, a 6-carbon acid called citric acid. And our job now is to go ahead and destroy that acetyl group, strip it off little bit by little bit. Now we need to go ahead and do this. If we want to strip off a carbon, we're going to have to do this as a Cu group. But our problem is, to get that Cu group, we only have 1 original oxygen left. So we're going to have to borrow oxygen from some source. Well, that happens to be water. We're going to go ahead and look at a couple of water molecules here. We're going to take one of those oxygens from each water, so I'll have 2 little oxygens here. And if we pump that into the system, we have enough to get rid of 1 carboxyl from each acetyl group. And of course, you're left with these pesky hydrogens up here, so I'm going to put them up here. There's 4 hydrogens that we got to worry about later, but don't worry about those. There's a reason they're there, but we won't find out till a little bit later. Anyway, now we've got our Cu group. We can go ahead and oxidize this, and we'll end up with 2 CO2s and end up with a couple of hydrogens. The hydrogens can get picked up by NADH, or excuse me, NAD, and bam, we get NADH. Since our citric acid is missing the equivalent of a Cu, it's no longer citric acid. We still have our original 4-carbon compound from oxaloacetic acid, but we also have one of our original carbons from the acetyl group, 3 hydrogens, and then of course no more oxygens. Dude, we got a methyl group on the end, huh? And of course there's 2 of those. That looks a little better, all cleaned up. Here's our methyl group that we left off with, and of course it's no longer citric acid but alpha-ketogluteric acid. So we want to go ahead and attack this little methyl group. And we can't just remove the methyl group. Our rule of thumb, if we're going to get rid of a carbon, we got to do it as a carboxyl. But now we're really in trouble because we surely don't have enough oxygen left to do this. So if we want to go ahead and get rid of the carbon, we're going to have to pump even more water in, right? So in this case, we're going to put 4 molecules of water. That'll give us 4 oxygens. 4 oxygens will be enough to make help us get rid of 2 carboxyls. So we're going to put 4 oxygens in here, and now we can strip off 2 carboxyls. And same thing that happened up here is going to happen down here. We can go ahead and strip this off as a couple of CO2s and hydrogen. Hydrogen gets picked up by NAD. Well, actually, there's 2 hydrogens, right? And 2 NADs to make 2 NADH. I'm sure you can all read this. And we're left off with our now 8 mystery hydrogens over here. So fear not, we're going to take care of that. In this whole process, when those carboxyl groups are stripped away, we get a couple ATPs out of it. Cool. Now we have, instead of alpha-ketogluteric acid, we've got succinic acid. And succinic acid still has everything that we had in our oxaloacetic acid. We still have our same original compound. But we end up with, in essence, now 2 hydrogens left over. And of course there's 2 of these. Okay? And if we start looking at what we're getting out of this, well, we had 2 CO2s, plus 2 CO2s, plus 2 CO2s from our pyruvate conversion. That's 6 CO2s. That's the same amount of waste that we're supposed to have in cellular respiration. So we've accounted for all of our CO2s. Right? Hydrogens, well, there's 2 carried here. There's 2 more carried down here. We had 2 from the glycolysis and 2 from pyruvate conversion. That's 8. And, well, we still got 4 more to deal with. And, shoot. We've still got these mystery hydrogens over here and these mystery hydrogens over here. Don't forget about those. They'll come into play a little bit later. Dude, that looks so much better when it's cleaned up. Alright. So we left off on succinic acid, and we've got just a couple of hydrogens that existed from original glucose. So our goal now is to go ahead and strip off hydrogens. We don't have to worry about putting any oxygen into this anymore, borrowing any oxygen from water, because We don't have to make any more carboxyl groups. There's no carbon left from that original molecule. All we got to do is strip off hydrogens. So in our next set of reactions, all we're really going to do is turn around and strip off a hydrogen, one hydrogen from each little fragment here. So I can get rid of 2 hydrogens. And it would be a wonderful, easy, and simple world if we could just have NADs come and take care of those. But for reasons that we are not going to go into here, NAD doesn't work in this step. We need a slightly different hydrogen and electron acceptor. That's where FADH comes in. And of course we need 2 of these. And we'll end up with 2 FADH2s. And get rid of one of our last remaining hydrogens. But succinic acid now has been converted to something called fumaric acid. And fumaric acid still has its 4 original carbons and all the other things that were attached to these that oxaloacetic acid has, except it also has an extra hydrogen. And of course, there's 2 of these. So our last step is, hey, we're just going to go ahead and go from fumaric acid compound back to oxaloacetic acid by getting rid of that hydrogen. And once again, our cleaned up version. So we want to go from here to here, but we got to get rid of that last little hydrogen. So again, enzymatically, we're just going to go ahead and strip that puppy off. Okay. That last hydrogen is going to be released, and of course there's 2 of them because we've got 2 fragments. And we'll go back to NADs. NAD picks up that hydrogen, and we end up with 2 reduced NADs, 2 NADHs. We're back to where we started. We haven't really changed that portion of the molecule, maybe rearranged some bonds, but we still have the same amount of carbons, hydrogens, oxygens that oxaloacetic acid actually started with. And it can be used over and over again to participate in this series of reactions. And we can have this cyclic cascade known throughout the world as the Krebs cycle, the citric acid cycle, the tricarboxylic acid cycle. Isn't life beautiful? What do you think? Can you trace your way through here? God's honest truth, I'm going to be honest with you. You don't necessarily have to remember all of the names of these compounds and events of these compounds to pass the advanced placement exam. What is important, though, is the thought process behind all of this and your ability to see how glucose is actually broken down kind of step by step. Now the problem with the process up to this point is that, yeah, we've created our CO2s, we've broken down glucose, and we've got a little bit of energy out of it, but dude, how much energy did we really get? We had 2 ATPs from glycolysis, none from pyruvate conversion, 2 ATPs from the rest of Krebs, and 4 ATPs is not enough to keep you alive. We did get our 6 CO2s, and although we used oxygen, we essentially borrowed oxygen from water molecules. Cellular respiration, you're supposed to use oxygen, free oxygen, some O2. We haven't really pumped that in yet. Well, that happens in the next step. We'll use our oxygen, and we'll get a bunch more energy out of it. Oh, I guess I really should show you the cleaned up version, so here it is. So yeah, take a mental snapshot of that. And yeah, if you ever want to explain the citric acid cycle to someone, like on a test or a quiz or something like that, wouldn't it be cool to actually explain your way through that? I'll bet your biology teacher would give you the blessings of many, many points if you were able to do so. Hint, 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 hint. Hey guys, that was a hint. Okay, our last set of reactions is where a lot of the magic happens. And it's a linear cascade series of reactions called the electron transport chain or the electron transport systems, or just ET. It also occurs in the mitochondria because things have to be shuffled through the membrane here. In order to go ahead and do this, we're going to use all those NADs, the reduced NADs and FADs that we gave off in the earlier sets of reactions. Those were not waste products. They're carrying the hydrogens, but more importantly, they're carrying high energy electrons that can be used to go ahead and do some special jobs here. Now, how are we going to convert electrons to energy? It happens all the time. Think what happens in electricity. We get electrons to flow through a wire, and we can power lights, we can power motors and engines. Here we're getting electrons not traveling through a wire, but carried by molecules. So yeah, there's a lot of power in electrons. We also need to see what happens to our mystery 12 hydrogens that we had in the citric acid cycle. And we promised that we'd be using oxygen. So this is going to be a true aerobic process. We're going to be using free oxygen molecules in the form of 6O2s. Now we're going to release so much energy that we're going to be able to phosphorylate 26 to 28 ADPs into ATPs. So how that actually happens, it's going to sound like magic at first, but trust me, I'll show you how it works. Anyway, NADs and FADs are going to be oxidized back into just NAD and FADH ions. We're going to need all that oxygen to actually accept all of the hydrogen and take that away from the system. Thus, we're going to go ahead and have water as a waste, but we end up with a little bit more water than we would think of in the summary equation. So let's see how this kind of the phosphorylation process of ADP and ATP is going to happen because of charged particles moving across membranes. So imagine hydrogen ions being shuffled across the mitochondrial membrane, and we're going to have a special channel protein called ATP synthase. I'll show you how this works in a sec. But as long as this flow of hydrogen ions continues through the channel protein, we end up with what is essentially a chemiosmotic gradient, allowing kinetic energy to convert ATP into ADP. And as long as we have this flow of ions continue, we can make more ATP. This requires that we'll have to go ahead and continually create a condition where there is always a high concentration of hydrogen on one side, lower on the other. So we have to continually remove hydrogen from one side. That's where oxygen comes in, pulling hydrogen away and keeping the concentration of hydrogen ions low on that side. I'll show you that in just a sec. Now to get where we're going, we have to go ahead and kind of sort of remove the hydrogens from NAD, essentially oxidize this or reoxidize NADs and FADs. So we're not going to go through all the reactions. So let's just say there's a series of protein complexes. We'll just identify one as complex 1. It's going to handle NADs to produce ubiquinone, and complex 2 is going to handle FADs. So NADs and FADs are going to be oxidized. The hydrogens are going to be stripped off. The electrons are going to be released. Right? Well, complex 3 is going to accept those electrons from both ubiquinone products to produce an intermediate product, cytochrome C. And this gets fed into complex number 4, and it's going to use those products to help oxygen go ahead and join with the hydrogen. So I don't think this process is as important as seeing ATP synthesis, so we're going to look at that. But before we do, research has demonstrated that for each FADH that you use, or FADH2 that you use in this process, you can generate about an ATP and a half, 1.5 ATPs, just on average. But remember, you have two FADHs, two FADH2s generated from Krebs cycle. Well, of course, 2 times 1.5, that is going to give you 3 ATPs, right? Well, NADH, you can get a little bit more ATPs out of those, 2.5. But remember that we got 2 of these NADHs from glycolysis, and 8 of them from pyruvate conversion and Krebs cycle. Well, 2 times 2.5 is 5, right? And 2 times 8 is, or 8 times 2.5 is 20. Right? So, hey, 20 plus 5 plus 3 is 28. We can get 28 ATPs generated through this process if we shuffle them through electron transport. Why do we say sometimes we only get 26? Well, remember that both these and these are already in the mitochondria. These were essentially released and generated out in the cytoplasm because that happened through glycolysis. So sometimes you need a little bit of energy to get these across the membrane. Think active transport. How much energy? A couple of ATPs. So, yeah, it's still worth it to bring it across. You still get a profit, but you don't get as much. So if we go ahead and spend a couple of ATPs, right, there's a possibility of only getting 26. But, hey, what the heck? That's still a whole lot more than we got from glycolysis and Krebs cycle by itself, so electron transport is still worth it. So here's how the ATP is actually generated. Through the various protein complexes, we're going to be able to take NADH and FADH2, pump them into this system, and essentially strip off the hydrogens and their attendant electrons. Right? This gets converted back into NAD and FADH, and it can be used and reused again and again in glycolysis, citric acid cycle, whatever. What we're left with is a bunch of hydrogens on this side of the membrane. Well, also embedded in the mitochondrial membrane are special channel proteins called ATP synthase. And these channel proteins are designed to go ahead and let hydrogen through. Remember, since this is a charged ion, it's not going to get through a membrane very easily unless you have some type of transmembranal help. Right? So in this case, here's our channel protein that allows that to occur. Now there's actually a physical rotor-like apparatus that helps convert this chemiosmotic charged almost electrical flow and convert this kinetic energy into energy that can be formed to make chemical bonds or used to make chemical bonds. Enough energy to go ahead and phosphorylate ADP plus an extra phosphate into ATP. And as long as this flow continues, that can happen. But imagine what's going to happen over time. We're eventually going to have a buildup of hydrogen ions on this side of the membrane. And you know from your studies of diffusion, when we have a equilibrium develop between concentrations on both sides of the membrane, this flow tends to actually stop, or the net flow stops. So if we no longer have a net flow of hydrogen ions from point A to point B, then production of ATP eventually stops. So as more hydrogens accumulate here, we have to have some way of getting rid of them. Well, why not just let oxygen do its job? If we pump oxygen in here, or allow oxygen to accumulate on this side of the membrane, then oxygen is going to combine with hydrogen, and eventually we'll end up with water. This can exit as a waste, and we end up with fewer and fewer hydrogen ions on this side of the membrane, and we always have a higher concentration of hydrogen on this side of the membrane. The flow continues from high concentration to low concentration, and we continue to go ahead and make ATP. Alright, so there you have it. You're going to need oxygen on this side of the membrane to continually remove hydrogens, and we can get this water as a waste. This ensures we always have a higher concentration of hydrogens on this side of the membrane, and thus we'll always go from high concentration to low concentration. We'll continue that flow through ATP synthase, and we'll guarantee that we're always creating lots and lots of ATP. Certainly enough to ensure that even complex multicellular organisms are going to be able to survive. Well, yeah, we need oxygen to live. But how much oxygen? Well, we were saying that for each molecule of glucose, you have to process or input 6 O2s. That's what our original equation said. But if we look at it, remember that we had 10 NADs going in, 2 FADs going in. That's 12 hydrogens that we added in. And if we wanted 12 hydrogens, we'd end up with something like this in terms of our water output. But if you look at the oxygen, it doesn't add up. Here, we got 6 oxygens in 6 water molecules. But we inputted 12 oxygen atoms. Where's the extra 6 oxygen? Where do they go to? Well, you might recall that in the citric acid cycle, we also had those 12 extra mystery hydrogens from the water that we inputted up there. So, lo and behold, if you take the 6 extra oxygen atoms that we have down here, add them up here, we end up with another 6 waters. And we can strip away those hydrogens. We've taken care of that. We can take care of our extra oxygen. And yeah, theoretically you have like 12 water molecules as a waste. But hey, if you do the math, it all figures out. And again, this is a lot like a mathematical equation. Right? If we go back to that original summary equation, we'll see that if we add 6 waters to each side, we still have an equivalent equation. We just kind of sort of simplified it. Anyway, play with it, have fun with it, and if you have questions, once again, hopefully you've been writing them down. If you get lost along the way and can't retrace your way back through all of the steps, we're going to go over it again in class, and hopefully we'll be able to go ahead and work things out together. Anyhow, I guess I'll see you in class next time. You have a great one, and yeah, enjoy your studies of cellular respiration. I know I will.